So this is going to be part 3 of your video, and in this video we are going to move on and talk about translation. So we just finished talking about transcription and how DNA is used to make an mRNA, and in this lecture we're going to talk about how mRNA is then used to code for protein. And so I have a question for you, and once I read it, go ahead and pause it, think of your answer, and then when you're ready, go ahead and push play again. And so the question says, an organism's genetic information is stored within the sequence of blank. This information is transcribed into a sequence of blank, which are then translated into a sequence of blank. Is it red, DNA bases, amino acids, RNA bases? Yellow, RNA bases, DNA bases, amino acids. Green, amino acids, DNA bases, RNA bases. Or blue, DNA bases, RNA bases, amino acids. So go ahead and pause. Okay, now, did you say that the answer is blue? If you did, then you're correct. Okay, and so the information is stored within DNA, which is transcribed into RNA bases, which is then translated into amino acids. And so this blue is the correct answer. Now, remember that we said that for eukaryotic cells, organisms that have a nucleus, transcription and translation are spatially separated. In the nucleus, remember, in eukaryotic cells, that's where you're going to find the DNA, and that in the cytoplasm is where translation is going to occur. And we mentioned that because they're spatially separated, RNA actually undergoes processing before it exits the nucleus. And so what does that really involve? One of the things that happens during RNA processing is that a 5' cap is added and a 3' tail is added. And the cap and tail serve a variety of functions. They help with the stability of the mRNA. They also help with starting and initiating translation. And another sequence that you should be familiar with is that if you look here on the DNA, here's the exon, here's the intron. Now what are those? Exons are the express sequences. These are the ones that actually code for the protein. Introns, on the other hand, these are called the intervening sequences. These are the sequences that need to be cut out, and they do not code for the protein. And so when you look at the DNA, you're going to see exon, intron, down the gene. During transcription, RNA polymerase is going to read the entire DNA. It's going to transcribe both the exons and the introns. And so it's going to make this primary transcript. And again, the primary transcript is going to have both the exons and the introns. But the RNA needs to be processed. And so it does something called splicing. And splicing is going to remove the introns. And so a structure called the spliceosome is going to come in. It's going to recognize these junctions between the exons and the introns. And it's going to loop out the intron and put the exons back together. And so now you get your mature mRNA. And your mature mRNA has all the coding sequences directly next to one another. We have now removed all of the introns. And now that mRNA is ready to exit out the nuclear pore and go into the cytoplasm to do translation. And so we're going to talk about how we go from RNA to protein. So, in order to understand translation, we need to talk about another type of RNA. And this type of RNA is called a transfer RNA. And a transfer RNA acts as the interpreter. It's the one that's going to read the mRNA language, it's going to read the codon, and it's going to bring with it the appropriate amino acid. If you look at the structure, here's the structure of a tRNA, and you'll notice that it has something called an anticodon. And what is an anticodon? So let's write something out real quick. So let's say that my DNA sequence is going to be let's say ATC. Okay, so this is just some made up DNA sequence. Let's say that my DNA sequence is ATC. And let's say that this DNA is going to be my template strand. If this is my template strand, that means that my mRNA is going to be complementary. So A is going to pair with U, T is going to pair with A, C is going to pair with G. So this is my mRNA, and that mRNA is going to be your codon. Now, if you look at the tRNA, this means that the tRNA anticodon is going to be complementary to the mRNA codon. So if this is a T, or I'm sorry, if this is a U on tRNA, it's going to be an A. And if on mRNA it's an A, it's going to be a U. And if on mRNA it's going to be a C, the tRNA is going to have, or I'm sorry, if the mRNA has a G, tRNA is going to have a U. And so the anticodon is on the tRNA, and the anticodon is going to be complementary to the mRNA codon. Okay, and so notice U is going to have A is going to have U, G is going to have C. And so this is the way that the tRNA is going to act as the interpreter. It's going to read the mRNA codon, and it has another important site that's called the amino acid attachment site. And this is where the amino acid will bind to. And so in this way, the tRNA is able to bring in the appropriate amino acid based on its anticodon, which recognizes a very specific codon on the mRNA. Now, remember that when we talked about cell organelles, we mentioned that the ribosome is responsible for protein synthesis. And so the ribosome is going to be responsible for translation. Remember back to when we talked about the ribosome, it has two subunits, a small subunit and a large subunit. And each subunit is made up of ribosomal RNA and proteins. And so remember we talked about that the nucleolus did ribosomal RNA synthesis, and it was also responsible for ribosome assembly. And so remember that the ribosomal RNA made the nucleolus, the proteins are going to come into the nucleus, ribosomal RNA is going to get put together in the nucleolus with them, and it's going to make our large and our small subunits. And the large and the small subunits don't come together until translation. And so the nucleolus is only responsible for synthesizing or putting together, assembling the large subunit, and it's also responsible for assembling the small subunit. But it doesn't put them both together until translation. The large and small subunit will come together in the cytoplasm. Now, an important feature of the ribosome is that it has binding sites for both mRNAs and tRNA. And so you could see that it's going to bind the mRNA, and it also is going to be able to bind the tRNA to bring the anticodon with the codon. And notice here you can see that this has two tRNAs bound together side by side. And we'll talk about how translation works and how we can add on an amino acid to a growing polypeptide chain. So, just like transcription, translation has 3 steps: initiation, elongation, termination. So the same 3 steps that transcription has. So let's start by going through what happens in each step. So in initiation, the first thing that's going to happen is that the mRNA binds to a small ribosomal subunit. Okay, so here we see, here's our mRNA, here is our small ribosomal subunit, and the small subunit is going to bind to the mRNA. Now, remember that when we do translation always starts, the start codon is AUG. And so notice that this sequence here, this we call the UTR. UTR stands for untranslated region. Notice there's no AUG. So translation doesn't begin until the first AUG. And that first AUG is going to be bound by a tRNA that has a sequence of UAC, right? It's going to be complementary to the AUG. And it's going to bring with it the amino acid methionine. Remember that all proteins initially will start with methionine as the first amino acid. And so once that tRNA docks at the mRNA, now the large ribosomal subunit will come in. It's going to bind to the small subunit, and now we have initiated translation. We have our functional ribosome, and now we're ready to translate the mRNA sequence. Elongation, right, is going to be where we elongate or grow our polypeptide chain. And so what's going to happen is remember that we said that for this ribosome that there are two spots where the tRNAs can bind. And so the next tRNA is going to come in and it's going to recognize the next codon. And so notice that your next codon here is GGA. And so a tRNA that's complementary that has CCU is going to come in and bind. Now, the important thing that's going to happen then is when these are coming together on the ribosome, those ribosomal RNAs are actually catalytic. They're able to catalyze or speed up the peptide bond between methionine and the next amino acid, so in this case glycine. And so it's going to form that peptide bond between those two amino acids. Methionine is going to leave its tRNA and it's now going to be on the glycine. That tRNA is now going to leave, and then the glycine is going to shift. Okay, and so the ribosome is like a ratchet. It's now going to shift down to the right, so it's going to go this way. That tRNA is going to leave, so here you can see the tRNA leave. And then now there's a new spot that can be occupied by the next tRNA. And so that next tRNA comes in. It's going to catalyze the peptide bond between these amino acids. Once it does that, the chain is now going to be over here. This tRNA is going to leave, and the next one's going to come in here. And so this is the elongation. This is going to be growing and lengthening that polypeptide chain. And then termination happens when we get to a stop codon. And so when you get to a stop codon, remember that there are 3 of them. The stop codons don't have a tRNA that's going to bind. Instead, they have a protein called the release factor protein. And this release factor protein is going to recognize the stop codon. It's going to bind, and because there's no amino acid to be brought with it, that's going to signal the end of translation. And so once we get this release factor and there's nowhere to add on to the polypeptide chain, now the ribosomal subunits are going to dissociate, they're going to come apart, and your polypeptide is now released and is fully formed. So how fast does translation occur? And the answer to that is that when we do translation, it's not that just one ribosome is going to bind at the beginning of the mRNA and it's going to go all the way through, and then it's going to come back and do it again and again. Instead, many ribosomes bind along the length of an mRNA, and they form something called the polyribosome structure. And so here's an electron micrograph. You can actually see this. So here you have your mRNA, and all of these little dots, those are going to be your ribosomes. And so you can see that there's many, many ribosomes along the length of an mRNA. And so because of this, because they're going to just kind of move along the mRNA, this is going to allow translation to be very, very fast. E. coli, which is a common bacteria that can be found in your gut, E. coli can string 40 amino acids per second. So an average size protein of 400 amino acids is built in just 10 seconds. Another example that really shows you how fast this is that a human immune cell, so your B cells, can manufacture 2,000 identical antibody proteins per second. So just think about that for a minute, how fast that is. It's like, boom, it just made 2,000 antibody proteins. So the translation is very, very, very rapid. And the reason that your immune system is so efficient is that you have these cells that are called memory cells. And your memory cells recognize a foreign invader that they've already seen. And once they see that, they have that information stored so that if you come in contact with that again, your immune system just cranks out antibodies to fight off that foreign invader. And so this is why you can't get sick with the same thing again and again. For example, right, if you've ever had the chicken pox, you know that once you have the chicken pox, you're not going to get it again. But if you think about the flu, right, you've probably had a flu many, many times. And that's because what ends up happening is the flu virus actually can mutate and it changes its shape. It's like a chameleon. It changes its shape, and so the antibodies that normally would recognize it, because its structure is a little bit different, those antibodies won't recognize it, and the flu is kind of just hiding out, and your body has to figure out new antibodies to produce. And so just something to kind of think about how amazing your bodies actually are.
Why?
So we talked about in our organelles lecture that for proteins that are going to be secreted, proteins that are going to be secreted will be made by bound ribosomes. Now, all ribosomes start out as free ribosomes, and they're going to be found in the cytoplasm. And so we said that during translation, that when we initiate translation, that the large and the small subunits are going to come together. And so when we start translation, the large subunit is going to be reading the mRNA, and it's going to make its amino acid chain. Now, if a protein is destined to become secreted, it has a sequence of amino acids at the beginning of the sequence that is what we call the signal peptide. And that signal peptide tells the cell where that protein is going to go. So what's going to happen then is that an SRP, this signal recognition particle, the SRP is going to recognize that signal peptide, and it's going to bind. And when it binds, it allows this ribosome to then dock onto the endoplasmic reticulum. And this SRP is going to bind to an SRP receptor protein. And that's going to allow the ribosome to dock onto the endoplasmic reticulum. And then the endoplasmic reticulum, the protein is going to be synthesized into the endoplasmic reticulum. The signal peptide is going to be cut off, it's going to be removed. And then your protein is going to be made inside the lumen of the ER. And so remember, I said that all proteins are going to start with methionine as the first amino acid, but they might not all end up with methionine as the first amino acid because it might be cut off by the endoplasmic reticulum. So here is a video on protein synthesis.
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured. The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA. As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus. The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases called an anticodon that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids. For proteins that will be secreted from the cell, like insulin, the ribosome docks on the rough ER, and the protein grows into the ER compartment. The new protein molecules are packaged in a vesicle that is transported to the Golgi apparatus, where many proteins are processed. However, insulin is packaged in a vesicle that leaves the Golgi and is then processed. Proteins secreted from the cell are shipped to the plasma membrane. Here, insulin is secreted from the pancreas and begins to regulate the student's rising blood sugar levels.
And so a question for you. A question says that the RNA that is translated into a polypeptide is blank RNA. And so red is nuclear, yellow is ribosomal, green is transfer, blue is messenger. So go ahead and pause it. Okay, think about your answer. And when you're ready, go ahead and turn it back on. So if you said blue, messenger RNA, you are correct, right? So the RNA that's translated into the polypeptide is going to be your messenger RNA.